Hello, my name is Kirill Shmilovich, and on behalf of my colleagues and collaborators, I'll be presenting work today entitled Discovering Molecules with Selective Membrane Partitioning Using Active Learning and Alchemical Transformation. So, mitochondria are the powerhouse of the cell. They are composed of two membranes, an inner mitochondrial membrane and an outer mitochondrial membrane. Oxidative phosphorylation for ATP production occurs throughout the inner membrane and intermembrane space. The outer mitochondrial membrane contains a large channel-forming protein called porin, which is permeable to all small molecules under 5,000 Daltons. The phospholipid cardiolipin is exclusively found in the inner mitochondrial membrane in eukaryotic cells and is involved in a number of metabolic pathways. Specifically, a number of diseases are related or linked to the precise composition of cardiolipin within the inner mitochondrial membranes, where cardiolipin abnormalities have in fact been linked to a number of diseases such as Barth syndrome or liver and heart disease. Molecules with selective affinity for cardiolipin could be used as a diagnostic tool for probing abnormalities in the pool of cardiolipin in vivo. In the past, the molecule 10N-nonyl acridine NaO, was used as a cardiolipin dye due to its high affinity for cardiolipin. However, it has been shown to be nonspecific while also associating to cardiolipin deficient cells. Therefore, the goal of this work is to identify molecules that selectively partition into cardiolipin-based membranes while remaining impermeable to other membrane phospholipids such as POPG. We engage this problem using active learning directed molecular design, and on the right is a schematic illustration of our molecular design pipeline. In brief, the first step is effectively the forward problem. We're given a molecular structure of interest, define a series of experiments or calculations, so in this case performing free energy calculations, that allow us to evaluate the performance of that molecule. The second step explicitly defines the design space that we elect to work in, importantly, framing the discrete molecular design space into a continuous representation that we can then directly expose to off-the-shelf optimization algorithms, namely using Bayesian optimization in steps 3 and 4. This pipeline is frequently called active learning, as it involves actively adapting the model and the data gathered iteration by iteration. I'm now going to describe the design space that we elect to work in for this work. So ideally, we'd want to consider all small organic molecules. However, this space becomes combinatorially large very quickly. So rather, we can consider a Martini-like coarse screening of all small organic molecules that involves only 5 different bead types. And in doing so, we're simultaneously able to accelerate the simulations that we perform, in effect, increasing the throughput of our technique, while also increasing the effective coverage of our design space while reducing our search space. Specifically, if we consider all different coarse-grained topologies with up to 5 different beads, so this corresponds to around 100,000 different topologies, this actually maps back to all molecules that are under around 400 Daltons or up to 10 to the 11 different unique atomic structures by only considering a coarse-grained design space of around 100,000 molecules. I'll now go on to describe each step in our active learning pipeline. So the first step involves performing free energy calculations of membrane selectivity. So for each molecule of interest, we perform a series of alchemical transformations in a number of different environments in order to estimate the selectivity of each molecule to cardiolipin. Specifically, we perform calculations in water, at the interface of the membrane and water, and in octane, which is meant to mimic the conditions of the bilayer midplane. Ultimately, we aim to minimize the difference of the free energy difference between water and the interface in cardiolipin and in POPG. In doing so, we are aiming to find a molecule that is preferentially inserts into cardiolipin, but not POPG. So we elect to use POPG here because POPG is chemically and structurally the most similar to cardiolipin. In principle, we could perform this calculation for each different phospholipid bilayer. However, this would be very expensive, and rather, we suppose that the if a molecule is found to be selective to cardiolipin over POPG, it is also likely to be selective to cardiolipin over other phospholipid bilayers. The next step involves creating the chemical space embedding, which we will ultimately perform our optimization within. So in order to do so, first we convert the coarse grain representations of our molecules into abstract graph objects that we will use in our deep learning setup. So specifically, each coarse grain molecule is represented as an abstract graph object with nodes, edges, and connectivity. The nodes are featurized according to the identity of the beads. So in this case, one of 7 different types of beads that are defined in this coarse grain force field. The adjacency, or the connectivity of the molecule, is defined through a binary adjacency matrix. And then the edges of our graph are featurized according to the strength of the pairwise Lennard-Jones epsilon parameter. So for this example, the edge connecting the T5 and the Q0 plus bead would be featurized according to the strength of the Lennard-Jones epsilon parameter between the T5 and the Q0 plus bead defined in the force field. We take these representations and feed them into a regularized autoencoder, which is a deterministic adaptation of variational autoencoders still capable of latent space interpolation and generative modeling. The input graph objects are processed using a graph neural network, which uses message passing to produce a latent code. That latent code is used to predict the reconstructed bead identities. And then together, those reconstructed bead identities and the latent code predict the connectivity of the reconstructed molecules. This together defines a effectively autoregressive T-coder where the connectivity and bead identities are used to reconstruct the original molecule. This whole network then is trained end-to-end, attempting to effectively reconstruct the input molecules while performing exact graph matching to ensure that our latent code remains invariant to permutational symmetries of the node identities. We can go on to interrogate this latent code in our model. In particular, the latent code that we use is actually a 16-dimensional object. However, we can view it in 3 dimensions by performing PCA on it. So here we see a 3-dimensional representation of our latent space, each molecule colored according to the number of beads in that molecule. So we see the molecules of 1, 2, 3, 4, and 5 beads are kind of distributed all throughout the latent space. Further, we can sample our latent space and confirm that we in fact produce a diversity of different topologies and bead identities by sampling the posterior of our latent space and decoding. We can further investigate how smooth the latent space is by performing interpolation between 2 points in our latent space. So specifically, we choose 2 points in our latent space and then linearly interpolate between them, where at each interpolate, we decode using our model and visualize the reconstruction. And we can see as we take small steps in our latent space correspond to small topological changes to the molecule. So for instance, the removal of just one edge or the changing of just one bead identity, where together we're able to smoothly interpolate between 2 target topologies in our latent space. The third step involves training a surrogate model to predict performance of unsampled points within the learned chemical space embedding based on measurements performed to date. Specifically, we use Gaussian process regression as our trainable surrogate model of choice, as it is a flexible non-parametric model that comes with built-in uncertainty estimates. The predictive accuracy of this technique is enhanced by the smooth, continuous, and low-dimensional nature of our chemical space embedding, which maps chemically similar points nearby one another to promote transfer of information to out-of-training points based on chemically proximate training examples. The last step takes the predictive estimates of the GPR to identify the next points to sample. This is done by evaluating an acquisition function, which inputs the predicted mean uncertainty from the GPR to select the next best points to sample based on balancing exploring undersampled regions of chemical space with large potential uncertainty and exploiting regions of chemical space with high predictive fitness. The fitness function is then evaluated on the candidates selected from this step, the outputs of which are reincorporated back into the training set, which is then used to refit the Gaussian process regression, and this whole loop continues until some desired performance is reached or some convergence criterion is met. So we've begun this sampling and have some preliminary results where we've sampled so far a total of 280 different molecules. So this constitutes 4 rounds of active learning, where in the first round we initialized our model with 100 randomly chosen molecules from the space, and then each subsequent round we select 60 molecules, we select 60 candidate molecules to simulate and to evaluate. So we can see that compared to the baseline 10N nonyl orange NaO, the currently used baseline, we've already sampled a number of molecules that outperform this baseline according to our free energy estimate of the selectivity of cardiolipin compared to POPG. Further, we can see that our model so far is improving round by round. So after each round has selected the next best candidate that has yet been seen. We can take a look at some of the best candidates that have been selected to date and begin to derive and think about some design rules that are coming up. So for instance, ring structures are very prevalent. So the ring structures in this coarse-grained space might correspond to different ring structures in all atom space, such as benzene rings. All the top candidates so far have a net positive charge and at least one hydrophobic bead. So this makes sense because the head groups of the cardiolipin are net negatively charged, and it's preferred for the molecule to be hydrophobic, which enables it to better insert into the membrane interface from the water. So some conclusions and outlooks. So far, we're performing high throughput virtual screening of small organic molecules using free energy calculations and alchemical transformations, and driven by an active learning directed Bayesian optimization. In doing so, we aim to discover new high performing candidates that are selective to cardiolipin over POPG, and then also derive design rules that can then be used to engineer these molecules. Some future outlook for this work is to, once we've finished a search in this coarse-grained space, we can back map some of the best candidates we've identified into all atom space for further verification, eventually passing the best candidates from that search on to our experimental collaborators that can directly measure the affinity and selectivity of these molecules to cardiolipin using by actually staining cells and measuring their selectivity and affinity. Some acknowledgements: my colleagues in the Ferguson lab, so my PI, Andrew Ferguson, and collaborators that we've been working with on this project, Dr. Tristan Bereau and Bernadette Mohr, and funding from the National Science Foundation Graduate Research Fellowship. Thank you everyone for your attention.